Hi there, thank you for watching the video of my NIRA poster. Connective soft tissue injuries occur at a high annual incidence in the United States, leading to the onset of other degenerative diseases and causing significant societal and economic burdens on society. The failure of these connective soft tissues is hypothesized to be caused by the damage to their collagen fiber matrix. However, current methods to detect this soft tissue damage are invasive and are infeasible in the clinical setting. Hence, we need to develop imaging methods that are capable of detecting soft tissue damage non-invasively. Diffusion tensor MRI is a promising non-invasive imaging technique that quantifies the diffusion of water molecules in a material. It is sensitive to physical barriers such as cells and fibers. Previously, we have found that in tissue mimicking fiber-based structures, fatigue-induced microstructural damage can be detected with DTI metrics, axial and radial diffusivity. Axial diffusivity measures the diffusion of water molecules along the main orientation of the fibers, whereas radial diffusivity measures the diffusion of water molecules perpendicular to the axial direction. These increases in axial and radial diffusivity are synonymous of microstructural fatigue-induced disruptions in tissue mimicking phantoms. Now, we want to translate these results into biological tissues and evaluate what is the effect of mechanically induced damage on the DTI metrics that we can measure in tendon, which is our research objective. Our hypothesis was that axial and radial diffusivity would increase in tendon as a result of mechanically induced damage. To accomplish our research objective, we use 5 deep digital flexor tendons from porcine feet. We obtained a DTI scan of the tendons before undergoing a fatigue protocol. Then, we mechanically induced damage of on the tendons using a fatigue loading protocol consisting of 10,000 loading cycles. After the tendons were damaged, we obtained another scans of the tendon post-fatigue. We also conducted a controlled DTI experiment on 4 tendons in which we did no mechanical loading on the tendons, but obtained 2 scans to obtain the baseline difference on our experiment. Of our tendon scans, we obtained 10 slices in which we analyzed 3 specific radial regions of interest: a central zone, a mid-radial zone, and an outer zone. Of each region of interest, we computed the diffusion tensor on each slice, and from the principal eigenvalue matrix of the diffusion tensor, we obtained the axial diffusivity, which is the first eigenvector, and the radial diffusivity, which is the mean of the second and third eigenvectors. Our first finding from our experiments was that the baseline changes in the control experiments result in decreased axial and radial diffusivity. Then, on our second finding, we see how by applying the mechanical loading protocol on the tendons, we had an increase in the mat cyclic displacement and a change in the stiffness of the tendons as a result of the mechanical loading. Now, when we looked at the pool data of the axial diffusivity, we have the probability density functions, which are equivalent to a weighted histogram of the data collected. We see that there was a significant increase in axial diffusivity on the mid-radial zone of the tendons. Then, when we looked at the radial diffusivity, we see a significant increase on both the central and the mid-radial zone of the tissues. We see no changes on either axial or radial diffusivity at the outer zone of the tendons. Then, our third finding shows how these changes in AD and RD are sample specific, which highlight the high sample-to-sample variability that is commonly observed in tendon biomechanical experiments. Here, we show the radial diffusivity measured for the 5 different tendons before and after the mechanical loading protocol on the central zone of the tissue. On sample 1, 2, and 4, we have a significant increase in radial diffusivity, whereas on sample 3 and sample 5, we don't have a significant difference, which shows how DTI can capture the sample-to-sample variability on tendons. Then, on our third finding, we can see that the changes in axial and radial diffusivity are location dependent. So, when we look at the change in radial diffusivity on the central zone, if we look at the data for the tendons on the proximal side or on slice 1, we see an increase in radial diffusivity on 4 of the 5 tendons. As we progress towards the distal size of the tendon on slice 10, we can see that there are no longer changes observed on the radial diffusivity. And when we look at the figures below that show the full field data of the local changes in radial diffusivity, we can see how on sample 2 specifically, on slice 1, we have higher overall changes in radial diffusivity that stops having these increases on slice 7 and 10, where radial diffusivity doesn't increase but actually decreases in some areas. What this data shows is that diffusion MRI can non-invasively detect mechanically induced damage in the tendons. So before fatigue, our DTI data shows that diffusion is restricted by collagen fibers or any other diffusion hindering structures in the tendons. And then when we look compared to the post-fatigue data, we see how these physical barriers or collagen fibers are disrupted and could potentially be either more disorganized or there could be more separation between them, especially on the proximal side of the tissue. And this can be captured using non-invasively using the diffusion tensor imaging data. Are conclusions that show that axial and radial diffusivity increase locally as a result of mechanically induced damage in tendon. We can also see how DTI metrics can be used to characterize heterogeneous damage progression in tendon and other soft tissues, and also how DTI is a promising non-invasive tool that can characterize damage and function in soft connective tissues, and that can be implemented in the clinical setting in order to address pressing needs in the treatment of connective soft tissue injuries. Thank you very much for watching this video, and please feel free to reach out to me if you have any questions. Thank you.